everyone, and welcome to today's webinar titled Curating the Genome to Drive Precision Diagnosis in Clinical Care. This webinar is part of the 10th Annual Genetics Virtual Week, and I'm Susie Valdez of Labroots, and I will be your moderator for today's event. Today's educational web seminar is presented by Labroots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. If you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the abstract window below the presentation window and follow the process to obtain those credits. Without further ado, I now present today's speaker, Dr. Mark Kiel, co-founder and chief science officer of Genomenon, and Catherine Nestor, Dr. Catherine Nestor, Vice President of Physician and Patient Strategies at Inozyme Pharma. For complete biography of our speakers, please visit the Biography tab at the left of your screen. Dr. Nestor, Dr. Kiel, welcome both of you. You may now begin your presentation.
Hello and welcome to our presentation at Labroots Genetics Week. I'm Mark. I'm the Chief Science Officer from Genomenon, and I feel so privileged to be presenting with a colleague at Inozyme, Catherine Nestor. We will be talking about curating the genome to drive precision diagnosis in clinical care for rare disease. My role is to introduce Genomenon very briefly and how we are curating the genome for this purpose, and to introduce Catherine, who's going to exemplify the value of this curation for a particular rare disease, ENPP1 deficiency. To begin with, I'd like to emphasize that Genomenon is a mission-driven company with a focus on making genomic information actionable to save and improve lives. Our focus is on the completeness and accuracy of this information for both clinical use, as I'll showcase, as well as in understanding the underlying genetic mechanisms of disease to help develop better drugs. The way that we deliver value into the clinic is through a genomic search engine that we refer to as Mastermind, which allows our clinical users to quickly identify references of genetic and genomic information to improve patient diagnosis and make more informed treatment decisions. Mastermind, the genomic search engine, allows users to search through genes and variants across millions of full-text scientific articles, millions of supplemental data sets, comprising many, many millions of genetic variants across the whole human genome. The software itself allows our users to analyze the results of that information in the context of those reference citations to provide rapid insight into the meaningfulness of that content. One of Genomenon's core capabilities we refer to as genomic language processing. This allows us to understand the information across these many resources in what I refer to here as the order from chaos, where the chaos on the left is the largely unstructured information and insight in these individual references at the full text and in supplemental data sets. In the middle there, you'll see all of the different entities that are genomic language processing indexes, from diseases and phenotypes and therapies, as well as genes and their genetic variants, no matter how an author may describe any of those pieces of information. In particular, the complexities of the genomic variant nomenclature is something that our GLP capability is expert at reconciling and organizing to maximize the sensitivity of a user search. When we index all this information, we put that content together in what we refer to as genomic associations. So the way that those genetic variants inform development of disease, the different clinical phenotypes that the patients might present with, and all of the different therapies that those patients were treated with and how they responded. That information put together is what gets displayed when a user searches on any one of those entities in the Mastermind Genomic Search Engine. An additional core capability that we have from a technical perspective that makes Genomenon unique is our expert curation capability that's informed by AI infrastructure internal to our team. So on the left, you can see the value of the GLP in ensuring the maximal sensitivity of indexing this information and pulling in that genomic association data that I talked about. On the right there, you can see exemplified our ability to ensure the maximal specificity of that information by arming our expert team of curators with this curation capability. And the fruits of that work allow us to make every assertion justifiable from any one of those reference citations that I spoke to earlier. What this culminates in for a select but growing list of genes that are associated with rare diseases and cancer is disease-specific curated content that we're now making available in the Mastermind Genomic Search Engine. What this means is that for every published genetic variant in one of those select genes, every one of those pertinent references that cite that variant and every relevant database entry necessary and sufficient to make clinical grade interpretations of the meaningfulness of those variants is presented to users after a provisional interpretation according to clinically accepted ACMG-AMP guidelines. What that looks like in practice, you can see here, where this is a screenshot of the Mastermind Genomic Search Engine, where at the top line for a given user search on one of those genetic variants and genes that I talked about earlier will be a summary of the fruits of that clinical grade interpretation. The pathogenicity or the way that those variants are associated with disease, and an itemized breakdown of the different categories of evidence that support that conclusion, as well as some action that those users can take to see that information in more detail and to be apprised of the steps that they can take to better diagnose and better treat those patients by enrolling them in clinical trials or by being made aware of commercially available drugs, depending on the circumstance. And one of the genes that we're very pleased to announce is in the Mastermind Genomic Search Engine is the ENPP1 gene, which is associated with ENPP1 deficiencies, which I'll pass the ball over to Catherine to present more clinical information on. Catherine?
Great, thanks, Mark. As Mark mentioned, when we set about our work in ENPP1 deficiency, one of the biggest challenges that we faced was how do we identify patients, how do clinicians get a more rapid diagnosis for their patients, and then how do we get these patients connected to the clinical studies. And so it was with this great need that we embarked on this work with Genomenon. And so before we get into sort of how all of this project came together, I wanted to share with everyone a little bit about the disease itself. So as Mark mentioned, ENPP1 deficiency is a disease that crosses the entire age spectrum. In the literature, and many clinicians know this disease by either GACI or ARHR2, and we'll talk a little bit more about those phenotypes as we move forward. This is a very heterogeneous disease, and there are some hallmarks of it that are helpful in recognizing it as clinicians are seeing their patients. Some of those hallmarks are ectopic calcification of the arteries, organs, joints, and ligaments. Patients tend to have pathological skeletal mineralization, and then also neointimal proliferation and vascular stenosis. So in a healthy population, what is the role of ENPP1? So ENPP1 plays a critical role in the regulation of mineralization and neointimal hyperplasia. And the mechanism by which this happens is that ABCC6 transports ATP from the intracellular space to the extracellular space.
And then EMPP1 cleaves ATP into PPI, and AMP CD73 further hydrolyzes AMP into adenosine and PI. So again, in a healthy population, what does this mean? Well, PPI maintains healthy mineralization. It inhibits the mineralization of arteries, organs, and joints, and maintains healthy teeth and bones. And then on the adenosine side of this, it maintains healthy vessel wall thickness. So in patients who have EMPP1 deficiency, what happens? Well, they don't have the enzyme to cleave ATP, and thus they have low PPI and low AMP, and then subsequently low adenosine. So clinically, the result of that is they mineralize, they have mineral deposits in arteries, joints, and organs. And then they also go on to have rickets and osteomalacia and reduced bone strength as the disease progresses. From the vessel side of things, this reduced adenosine level leads to neointimal hyperplasia and arterial stenosis. As I mentioned, it's very heterogeneous. If you look on the left of the slide, you'll see the infant presentation, and this is known again in the literature as generalized arterial calcification of infancy. It is really, these patients present with severe cardiovascular complications, and it has a very high mortality of more than 50% by 6 months of age. So for the infants that do survive that initial presentation and move into childhood and adolescence, the disease tends to evolve. FGF23 rises, and these patients tend to have hypophosphatemic rickets. They have bone deformities, impaired growth, and hearing loss. And if they survive into adulthood, they tend to have bone and joint pain and impaired mobility. And this phenotype is known as autosomal recessive hypophosphatemic rickets type 2. So I mentioned that it's heterogeneous, and I also mentioned the high mortality. This is a population from Dr. Roach et al., and he looked at a retrospective natural history study in 55 patients with GACI. 65% of those patients died either in utero or within the first 6 months of life. And as you see on the right here, the sites of calcification associated with death and survival in this population, significantly, the aorta was involved, coronary arteries, pulmonary arteries, etc. here. The most frequent signs and symptoms in the prenatal period are fetal distress, polyhydramnios, and pericardial effusion. Some of these babies will have fetal hydrops. And then in the neonatal period, as I mentioned, cardiac insufficiency, respiratory distress, arterial hypertension, and impalpable pulses. So for patients who survive that first critical period, as I mentioned, they go on to develop hypophosphatemia. Looking at 8 of 19 children who survived those first 6 months and for which we had serial measurements of serum phosphate, we do see that all 8 of them developed hypophosphatemia and hyperphosphaturia, which was first noted between the 2nd and 3rd year of life. As I mentioned, FGF23 rises in these patients, and as you'll see here, there were elevated FGF23 levels in relation to their serum phosphate levels observed in this cohort of patients. So let's talk a little bit more about the hypophosphatemic rickets that these patients experience. In a retrospective study looking at a total of 84 patients, 49% of those patients had signs and symptoms of rickets. You'll see on the left there a 7-year-old with bowing of both femurs. In the middle, you'll see an adult patient with bowing of both femurs. And then on the right, a 7-year-old male with metaphyseal fraying and flaring in the knee and ankle. So how do we differentiate EMPP1 deficiency from other forms of hypophosphatemic rickets? It can sometimes be a challenge. You'll see here the biochemical findings in children and adults with EMPP1 deficiency may be identical to other forms of hypophosphatemic rickets, such as XLH. Biochemically, these diseases can look identical, And so it really relies on genetic sequencing to make the final diagnosis. And we'll talk a little bit more about that in a moment. So if we look at the clinical features of both of those diseases, you'll see on the left there some of the keys to an XLH diagnosis. And then on the right, the keys to the ENPP1 diagnosis. In the middle, you'll see that there is some significant clinical overlap. So some findings that could help clinician, sort of lead in the direction of the ENPP1 deficiency, of course, are the arterial organ or joint calcifications, intimal proliferation, delayed onset of rickets, spinal ligament calcification, and then unlike the dental manifestations you would see in an XLH population, we see hypercementum or delayed shedding of teeth for ENPP1 patients. And then some other clinical features in some children and adults, calcification and or fusion of the cervical spine ligament. I mentioned the hypercementum and delayed shedding of teeth. Again, arterial calcification and neo-intimal proliferation, and then cardiovascular complications, which can continue into childhood and adulthood for some patients. So the diagnosis, it really does take a multidisciplinary approach, a really good medical, any family members who may have had signs and symptoms of ENPP1 deficiency. The clinical exam would be similar to what you would do for any skeletal disease. Radiographs, as I mentioned, you could see the rachitic changes. The hearing exam is important because hearing loss does happen commonly in kids with ENPP1 deficiency. You may see some retinal changes. You'll see, as I mentioned, the calcification on echo, including in some cases large vessels, and then the laboratory findings as well. But at the end of the day, the confirmation of ENPP1 deficiency does require genetic sequencing. So some pathways, some diagnostic pathways have been developed with input from some expert physician groups. This pathway is specific to the in utero and infant population. And as you'll see, it really is driven by the cardiovascular findings and echo brightness of the disease. If you can get serum PPI measures, those can also be helpful. These patients will have low serum PPI. But again, the ultimate decision maker here is going to be genetics. So that's infants and in utero. There's also a pathway developed for kids less than, greater than 1 up to age 16 years of age. As the disease evolves, the skeletal manifestations really become the driver of the clinical findings. And so this pathway is more focused on the skeletal findings of children, and then the cardiac findings still exist, but are less prominent in that population. And then for the adults, it really is about the bone findings, osteomalacia, in some cases, tumor-induced osteomalacia for which you can't find a presence of a tumor, treatment-resistant osteoporosis, or FEX-negative XLH are all clues to maybe this could be ENPP1 deficiency. So there is a clinical study for the disease. We've developed this product, INZ-701, and it's been designed to replace the lost enzymatic function of ENPP1. It is a recombinant human ENPP1 protein, and the construct for it is recombinant Fc fusion protein with a soluble extracellular domain of ENPP1. Right now, the study is in adults, and it's dosing 2 times per week subcutaneously. It's a phase 1/2. I'll share a little bit more about this study here. We just released yesterday cohort data from the first 3 patients in which the drug was found to be safe, and PPI was raised in all 3 subjects in the first cohort. So it will continue on to have 2 more cohorts in which it will be an ascending dose, and then finally a dose selection will be decided after all data is collected. And there's a long-term extension component to this study as well, for which patients will be followed for an additional 44 weeks. And that study is open and enrolling, as I mentioned. So with that, I'm going to turn it back over to Mark.
Thank you so much, Catherine. And just to emphasize what Catherine brought up several times during her presentation is that ENPP1 deficiency requires a genetic confirmation. And presently, that's challenged by an incomplete awareness of the causative variants in ENPP1, and it's especially challenged by the rarity of this disease and the clinical heterogeneity and overlap with other diseases. So genetics really gets at clarifying an ENPP1 diagnosis, and having the complete comprehensive understanding of those causative variants is really required in order to ensure optimal diagnosis across entire patient populations. So there's real power in the comprehensiveness and the accuracy of this curated information comprising all of these causative variants for any given gene like ENPP1. The value that that comprehensiveness provides, I'm illustrating here on this slide. Allow me to walk through it because it's a little complicated, but on the left is the scenario where you don't have a comprehensive understanding, but you still have the ability to designate benign, likely benign, variant of uncertain significance, likely pathogenic, or pathogenic status to certain variants in the gene. A comprehensive understanding that arises from curation of this information using GLP and the AI curation capabilities that Genomenon has that I talked to earlier does two things. It increases the awareness of those variants by making all of those bars rise, by providing a comprehensive understanding of the causative variants in that gene. But it also increases the evidence, the cases, the clinical information, the diagnostic relevance, the phenotypic information, as well as the functional studies demonstrating the disease-causing nature of any one of those variants. That increase in the evidence per variant will not only raise each of those bars, but it will also shift those bars to the left by providing more, to the right, by providing more of that information to ensure that variants that do cause disease meet clinical standards of evidence to promote them from variants of uncertain significance, where there's not enough evidence, to likely pathogenic or pathogenic. So the net result is having a more comprehensive understanding of the causative variant landscape and all of the evidence supporting those determinations will lead to a better understanding of those pathogenic variants, ultimately leading to higher diagnostic yield with more patients being diagnosed. The way that this information can be put to action is through the disease-specific curated content that we talked about earlier, where when a clinician or a laboratorian performs a search on any one of those variants, the evidence and the information that was part of that comprehensive understanding of all those variants is then presented to that end user for immediate actionability. In this case, example that we're talking about for ENPP1, if a clinical or laboratory user performs a search on one of those ENPP1 variants, the provisional interpretation of the meaningfulness of those that variant is presented to them directly, as you can see here in that red banner at the top of our software interface, as well as a categorization of the evidence that culminated in that interpretation. Clicking on one of the buttons there, the View Interpretation button, will actually show you the information in great detail, the annotated, categorized, and enumerated information that culminated in that clinical interpretation for that variant. And you can see here the database information off to the left at the bottom, as well as the evidence from those clinical and functional references that I alluded to earlier, all expertly curated, all tagged with the categories of relevance according to the clinical interpretation guidelines, the ACMG AMP criteria, for whether they're clinical, whether they're functional, lines of evidence that support the pathogenicity and what the relative weights of any one of those components of the evidence are, so that our work is shown when we've curated that information for the clinician and laboratorian to very quickly appreciate what evidence exists, what the categorizations has been, and to verify that they agree with those components so that they can promulgate that into their clinical report to ensure the utmost accuracy in their diagnosis, or as Catherine had alluded to on her last slide, be made aware of ongoing trials that are enrolling patients, as you can see here. So this is an opt-in approach for clinicians who see a patient with one of these causative variants. They may not be well aware of the trial that's ongoing, given the rarity of this disease. And that's one of the powers of disease-specific curated content being integrated into our clinical software, as you can see, in the service of maximizing the diagnostic rate for rare disease patients like those afflicted with ENPP1 deficiency. So to summarize, as I've indicated, disease-specific curated content is available in our clinical software now for a select but growing number of genes, including ENPP1. And what that comprises is every published genetic variant in any one of those genes, every single one of those variants, with every one of the references that cite those variants, expertly assessed and interpreted for meaningfulness by our team of variant analysts, master's level and above, as well as an interpretation of every relevant database component, such as population frequency and predictive models of pathogenicity. Those three components, the publication information, the prediction information, and the population frequency, are all interpreted according to those clinical standard frameworks and presented in the Mastermind interface for the clinicians and laboratorians to see the maximal extent of that evidence, to know what the provisional call was, and to be able to take action on that call by being notified of clinical trials and being made to have connections with the trial coordinators. So with that, I'll conclude, and thank you for attending our talk, where again we discussed how Genomenon is curating the genome to drive precision diagnosis in clinical care for rare disease, with a particular emphasis on one important rare disease, ENPP1 deficiency, by our colleagues at Inozyme.
Thank you, Dr. Nasser. Thank you, Dr. Kiel, for your time today and for your important research. And before we go, I want to thank our audience for joining us today. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until April 21st, 2024. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care.